Okay, I would like to take this opportunity to thank the organizers for giving us this opportunity to present our work. Okay, let's see. Okay, so as everyone is aware here, loss of function mutations in Tafazzin result in the development of Barth syndrome. And the most common clinical features of Barth syndrome include cardiomyopathy, skeletal myopathy, cyclic neutropenia, and growth deficiency. In addition, low levels of plasma cholesterol in the low-density lipoprotein fraction have also been reported. So growth deficiency occurs for both height and weight in most Barth syndrome patients. And so growth curves have recently been developed, and it is established that the 50th percentile for height of Barth syndrome patients, shown in red, is shifted down below the 10th percentile for the general US population. So indicating that the average height for Barth syndrome patients is significantly reduced. And in addition, the 50th percentile for weight of Barth syndrome patients, again shown in red, is shifted down below the 10th percentile for the general US population. So again, indicating that the average weight of patients is also significantly reduced. So in fact, the weight of almost 60% of patients is below the 5th percentile, and there are corresponding reductions in body mass index, indicating that this reduction in weight is irrespective of height. There's also been reports or a history of failure to thrive in 54% of patients. So together, these data or findings indicate that there is a skinny phenotype in some individuals who have Barth syndrome. So body weight is controlled by both food intake and absorption, as well as energy expenditure, and is reflected in the amount of fat mass or adipose tissue, as well as the amount of lean body mass, which is predominantly skeletal muscle, but also the heart, visceral organs, including the liver, and bone. So the purpose of this study was to determine how Tafazzin deficiency reduces weight gain in mice using the doxycycline-inducible knockdown. So for our experimental model, we used male mice, which contained the cassette, which had the short hairpin RNA directed against Tafazzin under the control of the doxycycline-inducible promoter and their wild-type littermates. So we have the animals which contain the cassette shown here and the wild-type littermates, and we divided them into 3 groups. So the first group was fed the regular chow-fed diet, so no doxycycline, such that Tafazzin is not knocked down. And this was typically for 4 months. And the other 2 groups were fed doxycycline diet for either 4 months or 10 months, and this was to look at the lack of weight gain in the mice over time. So I'll refer to the mice fed the doxycycline diet as the TAS wild type and knockdown. Okay. So the first thing that we wanted to do was to look at whether a skinny phenotype was actually produced in the TAS knockdown mice. And in fact, we found that the TAS knockdown mice were indeed smaller than their wild-type controls. We found that the TAS knockdown mice shown in yellow had a reduced growth curve compared to both the regular chow-fed mice shown in black and red, as well as the wild-type mice shown in green. So the TAS knockdown mice were significantly smaller than the regular chow-fed mice starting at 4 months of age, and they increased to a maximum of 25% reduction at 10 months of age. And so the reduction in body weight was more dramatic when we looked or compared them to the TAS wild-type mice. So the TAS knockdowns were significantly smaller starting at 2 months of age, and they increased to a maximum of 40% reduction at 10 months of age. So we presume that there was such a large increase in weight in the TAS wild-type mice because of the addition of doxycycline to the diet. So antibiotics have been shown to increase weight gain without additional calories. So how has the body composition been altered in the TAS knockdown mice to achieve this skinny phenotype? So we first looked at bone growth, and we found that there was no difference with respect to tibia length by age or genotype. And we also looked at bone mineral density and found that there was no difference between the different groups, and this was done by DEXA scanning. So the small size of the TAS knockdown mouse was not due to impaired skeletal development. So we also used DEXA scanning to look at the amount of fat, as well as the amount of lean body mass in the regular chow-fed mice, as well as the wild type and knockdown. And we found the wild type mice weighed more due to increases in both fat, as well as lean body mass compared to the regular chow-fed mice. And TAS deficiency was impairing this weight gain predominantly due to reduction in fat mass. This is at 4 months, and there was little difference between the genotypes with respect to lean body mass in between wild type and knockdown. So similarly, at 10 months of age, we saw a significant 76% reduction in fat mass between the knockdown and wild type, and there was a small but significant reduction in lean body mass between the wild types and knockdowns. So the TAS knockdowns were smaller due to a significant reduction in fat mass. And this result was confirmed when we looked at the size of various fat pads. So we saw significant 70% reductions in gonadal and perirenal fat pads, and then a 50% reduction in brown adipose tissue. And we saw this both at 4 months and also the same percentage decreases at 10 months of age. So a major contributor to fat pad mass is the expansion or hypertrophy of adipocytes to accommodate triglyceride storage. And so we looked at the gonadal fat pad histologically, and we saw that in the TAS knockdown mice, the diameter of the adipocytes was much smaller than that in the TAS wild type. And this was at both 4 and 10 months of age. So TAS deficiency was impairing triglyceride storage in the adipocytes. Okay, so is lipid accumulation also being impaired in other tissues? And we looked at skeletal muscle, and we found significant reductions of triglyceride in the TAS knockdown compared to the wild type at both time points, although it was more dramatically at the 10-month time point. And we weighed a number of tissues. So we weighed the heart and the pancreas and the liver, because a reduction in weight can indicate a reduction in lipid accumulation. And so similar with previous findings, we found increase in heart weight at 4 months in the TAS knockdown compared to the wild type, and increase in both pancreas and heart at 10 months. But we also saw this reduction in liver weight at 10 months of age in the TAS knockdown, which indicates a potential reduction in lipid accumulation. And so when we looked at the livers histologically, so these are H&E slides and oil-red-O slides, which stands for neutral lipid, we saw there was no change at 4 months between the genotypes. And then at 10 months, the wild-type livers were filled with vacuoles, which contain neutral lipid as determined by oil-red-O. And in comparison, the TAS knockdown mice, the livers from those animals, did not contain any of these lipid-filled vacuoles. So we confirmed this finding when we actually measured the amount of triglyceride in the liver. We found it was significantly reduced. And we also looked at another neutral lipid, which is cholesterol ester, and found that it was significantly reduced as well at the 10-month time point. So the accumulation of neutral lipids is reduced in the liver in the TAS knockdown mice. So since hepatic lipid metabolism also includes the synthesis and secretion of lipoproteins, we looked at plasma lipid levels. And interestingly, we found that the reduction in hepatic lipid levels in the TAS knockdown was also mirrored in the plasma. So we found that there was significant reductions in plasma triglyceride in the TAS knockdown mice. And we also used fast protein liquid chromatography, which separates lipoproteins by size. So we get very low density lipoproteins, low density lipoproteins, and high density lipoproteins. And so we can see that the reduction of triglyceride was in the very low density lipoprotein fraction. And we looked at plasma cholesterol ester levels. And similarly, we found reductions in plasma cholesterol ester in the TAS knockdown at both time points. And again, using fast protein liquid chromatography, we can see that the reduction in cholesterol, so this is the TAS knockdown in red, so the reduction in cholesterol is in the low density lipoprotein fraction, and there is little change in the very low or high density lipoprotein fraction. So that's similar to the clinical feature of Barth syndrome. Okay, so we've now established that the TAS knockdown mice have a skinny phenotype, and we have reduced adipose and liver tissue mass. We have reduced triglyceride levels in the adipose, muscle, and liver, and we have reduced plasma, VLDL, triglyceride, and LDL cholesterol. So we wanted to look at how TAS deficiency is preventing this weight gain. So the first thing that we did was look at food consumption. And so while the doxycycline-fed animals were consuming less food per gram of body weight in a 24-hour period of time than the regular chow, you can see that the TAS knockdowns were consistently eating more than the TAS wild type. So the TAS knockdown mice weren't smaller simply because they were consuming less food. We also looked at intestinal absorption, which is good because of the last, the last presentation, to see if TAS knockdown was having an effect on the absorption of calories. And so we looked at the kilocalories absorbed in a 24-hour period, and we found that there was no difference between the genotypes at either time point. Okay, so then we looked at energy expenditure using indirect calorimetry, and we found that the TAS knockdown mice shown in the open circles had increased oxygen consumption both during the day as well as during the night compared to the TAS wild type in the closed circles. And we can see this both in our scatter graph as well as the average, the corresponding averages for both the day and the night in the bar graph. And similarly, we found increased heat production in the TAS knockdowns, again shown in the open circles both during the day and the night, compared to the wild types. And again, we show the corresponding averages for both the day and the night, and we can see significant increases. So the TAS knockdown mice have elevated heat production and increased energy expenditure. So finally, we looked at the respiratory exchange ratio for our animals. And so this is a ratio of carbon dioxide generated per oxygen consumed. And so because fatty acids use more oxygen than carbohydrate to generate carbon dioxide, it gives you the proportion of each substrate being used. So we see that the TAS knockdown mice are proportionally using more fat than carbohydrate compared to the wild type mice, both during the day and the night. Okay, so to summarize, what we've observed is that there's the TAS knockdown mice have this skinny phenotype. It is characterized by reduced mass and triglyceride levels in the adipose, muscle, and liver. And there's reduced plasma, VLDL, triglyceride, and LDL cholesterol. And appears to be due to elevated oxygen consumption, elevated heat production, and we have a preference for fatty acid oxidation. So for our working model is that in the TAS knockdown mice, there's increased heat expenditure, which is driving up energy expenditure, and we don't see any change in food intake. And this is reducing the body weight. And this skinny phenotype is then predominantly due to a reduction in fat mass with less triglyceride in the adipose tissue. And then we have a smaller reduction in lean body mass with less triglyceride in both the skeletal muscle and also in the liver. And so at this time, we've concluded that we established that the mice have hypermetabolism and altered triglyceride metabolism, which is contributing to their low body weight. And so I would like to thank my supervisor, Dr. Grant Hatch, as well as various collaborators and excellent technical support by a number of individuals at the University of Manitoba as well as at the University of Alberta. And of course, our funding agencies, most notably the Barth Syndrome Foundation, also Manitoba Institute of Child Health and Canadian Institute for Health Research. And please take any questions. Thank you.
Thank you very much. That was a very intriguing talk.
Thank you. It's a very clear talk. In the UK, we have a different group of patients in that we find that they have a high fat mass compared with their height. And yeah, it's very interesting. I don't know quite why it's so different. And it's yeah, it certainly contradicts what you've been saying, but maybe it's just a completely different environment or different diet, or I don't know, but...
Well, one of the confounding, they show the growth curves for Barth syndrome patients, at least in the US. It seems that they tend to have a reduction in body weight, but that's not everybody, right? So if there's 60% of people have a low body weight, there's still 40% which don't. Also, our mice are fed doxycycline, which does confound some of the results. So, yeah.
Really interesting talk. Thanks so much. Just a quick question. When you normalize the indirect calorimetry data, what does it look like when you don't correct for body mass? Because you're talking about mice that are 25% different in weight, correct?
Right. We correct by lean body mass.
And on a per mouse basis, is it still significantly different? If
Well,
you just look at...
Well, it's going to be, I guess it depends on, yeah, so it won't be different, I'm not really sure, actually. I'd have to go back and look at the data. But if we correct by lean body mass, it's different.
It's different.
Yeah.
Cool, thank you.
So this is a follow-up to Anne's comment. So when we look at our patients, it's every time it's really the fat-free mass that's driving the low weight, and the fat does not seem to be any different. And sometimes it could be a little bit more, but it hasn't. Can you explain a little bit more why you think it's a model specific? Was there activity higher, or could this be explained by the increased brown adipose tissue, increased oxygen consumption that's limiting this fat accumulation?
Okay, so the first question was, do we have reduction activity?
In fat, fat-free mass.
Why do we have a reduction in fat-free mass?
No, do you see the reduction in fat-free mass in the mouse model? So what we see in humans is they have lower weight, but it's driven primarily by the reduction in fat-free mass versus fat mass. Right,
so I showed the fat-free mass, and we do see some reduction, but it wasn't as large as the fat mass. We have the activity data, and I haven't analyzed all of it. Initially, it looked like there might not be much of a change, but I need to crunch more numbers. So it could be contributing, although we do see a change in heat production. So regardless of if there's a change in activity, there's still an increase in heat production as well. So here's there was no change in lean body mass at the earlier time point, and then a smaller one at 10 months.
I wonder, while I'm standing here, if I can ask Todd about the respiratory exchange ratio in the patients and compared to this.
I can't really explain it. So what we found, so when you exercise, so at rest, they basically use the same amount of fat as their peers. And it seems like they use more glucose, which you're kind of finding the opposite. When you stress them, when you exercise them, they're not able to use fat as efficiently, and they seem to compensate with the glucose. But that's not oxidation that we're looking at. We haven't specifically used a 13C tracer, glucose tracer. So, but I'm trying to reconcile some of your findings. I'm not sure if it's an animal specific. It might be because of their increased energy expenditure and a little bit different that you're finding these body composition differences and also metabolic differences. But yeah.
Yeah, so I mean, you have to keep in mind too that the RER data is proportionally, so it's not an absolute amount. So it simply means that the mouse is using more of one substrate than the other. It doesn't mean that it's using more or less than the wild type. I need to do that quantitatively, look at the oxidation of each substrate individually to determine that. Yeah, but I do think that the doxycycline may be having effects on the animal, and there's reports that doxycycline impairs fatty acid oxidation. So also need to keep this in mind when interpreting the data and trying to correlate or make it comparable to human data. Yeah.
So it's a very nice talk. It's a little confusing because you think about cardiac and skeletal muscle, and they really aren't oxidizing fatty acids, but you're brown adipose or your white adipose are oxidizing fat clearly in your model. So is this simply due to a tissue-specific expression of Tafazzin, and then the brown adipose tissue, the tissue is perfectly normal because Tafazzin is not being expressed there?
Well, we also see less brown adipose tissue as well in the Tafazzin knockdown mice, so it would have to be
But it's very efficiently
right, but it still could
oxidizing
be,
fat.
It still could be oxidizing fat. We're also interested in what the liver is doing as well.
Have you looked at TAS mRNA levels in that tissue?
In brown adipose tissue, no, we haven't.
So that it seems to me that the only way to reconcile it is if the TAS expression is not occurring there, and we don't have the same defect in mitochondrial metabolism in that tissue.
Right, we could definitely look at that for sure.
Other questions?
So I mean, the mouse could be different from humans, right?
Right.
I mean, that's you know. So I wouldn't worry too much about that difference to humans, but because you still have something interesting to understand. So
Right.
if you see the combination of excess heat production and lean or skinny phenotype, and you immediately think it could be uncoupling of mitochondria,
Yeah,
right?
exactly.
So,
especially with respect to Edgar's results, which he showed earlier, that there
so
was.
you think that's a possible explanation?
Yeah, for sure. Yeah.
Just one last question. I guess in the patients, it seems that there's a growth delay during childhood. So I guess in the equivalent period in the mice, it's sort of compressed on your graph. But I don't know in that initial period during the initial growth of the mice before puberty, if there's any difference in these mice.
Actually, we don't really measure them too much before about 2 months of age. So we saw some change in body weight between the wild type and knockdown. So it was significant at 2 months of age. Oh, do I have this the right way? At 2 months of age between the TAS and wild type. But it's also partially because of the numbers that we have. So we may have 30 or 40 animals here, so that's why it doesn't look very substantial, but the power of the statistics is high there. So at 1 month, I haven't seen any difference, but there's many fewer animals that I've looked at this time point, and generally they're not part of our protocol until they're closer to 2 months of age. So I don't really know, I guess, yeah.
Okay, thank you very much. Let's have a hand. Oh, sorry. Barry has a question. Premature.
Is the doxycycline given in the chow or the water, and is it given to the dams?
Yes, so it's given to, it's in the food, so it's the 625 mgs per kilogram, and it's given to the dams, right? Okay. So they're exposed to it for their entire lives, so, or even before before they're born, at conception.
Yeah. Okay, great.
Any other questions?
Okay.
Oh, there is another question.
Are these still active as normal or wild type mice at 10 months of age?
Are they still active?
More active or less active than?
Well, I would say that the TAS knockdown are pretty active. The wild types are pretty obese, so they're not moving too much. Yeah.
Okay, thank you.